next portion. So Chris Schadt and Spencer Roth will be tag teaming our presentation on the SPRUCE experiment. So I'll hand it over to you, Chris.
Okay, well, thank you to the team for inviting myself and Spencer to go over some of the things we've been doing on the SPRUCE project. And it sounds like you will be getting familiar with this data set over the course of the week. So we can give you a little context, hopefully, for where this is coming from and why we're doing it. So the reason we're interested in gathering this data to start with is that peatlands and peatland soil systems really play a very large, comparatively large role in soil carbon cycling and carbon cycling in general across the globe. So despite covering very little of the surface area of the Earth at less than 3% or so, globally, peatlands store over 30% of all the soil carbon on Earth, which is quite amazing. Most of that carbon in these peatland systems is often located very deep relative from the surface. So we often think a lot of soil science is done in the top 20 centimeters or foot of soil, that is the plow layer traditionally. These are often very deep, rich organic soils all the way to depths of meters or more. And these, the responses of these deep peat deposits are important potential feedbacks to the climate carbon cycle. So as these deep peat deposits either stay or go away under changing climate, what kind of feedback that could have on the climate change is quite different. Oops. So give a little more context, the peatlands we're talking about are part of the boreal forest ecosystems in the United States and Canada. Those ecosystems stretch globally as well, but the SPRUCE experiment itself is located at sort of the southern end of that boreal ecosystem. So in northern Minnesota in the US. So this is showing the extent of that ecosystem across North America and the total tree canopy cover in green. So specifically, the SPRUCE experiment is located within the Marcell experimental forest run by the US Forest Service in northern Minnesota, north of Grand Rapids, Minnesota. So, and this is a experimental forest that has really been an active research site for over 50 years. So beginning in the 60s, the research has been going on since then. They have several intensive study sites at different bogs and fens within these systems. So for example, the S1 through S8 fen bogs have been studied quite extensively over the course of those years. Specifically, the SPRUCE experiment is located in what was called the S1 bog by the experimental forest. And these bogs were, each of these bogs were monitored as a watershed level, including the bog area, the wetland area itself in the hatched color, and the upland area as sort of a mini watershed for each of these major bog study areas, where they could understand export not only from the bog, but from the surrounding uplands into the bog on a watershed scale. So the SPRUCE experiment is a climate change experiment where we're warming these peatland ecosystems by up to 9 degrees Celsius, both above and below ground. So above ground, these consist of 12-meter diameter study area inside of a greenhouse-like chamber. Each of these chambers is So each of the 10 chambers here range in a regression design from zero energy added, so no heat added, just the infrastructure added, to plus 2.25 degrees Celsius, plus 4.5 degrees Celsius, plus 6.75 degrees Celsius, and plus 9 degrees Celsius. So in a regression design, we can look at the response surface of different environmental variables to this increases in temperature. And so above ground, enclosed by a greenhouse-like chamber with a center boardwalk. So the center area between the boardwalks being the primary study area. And below ground, you can't see in the pictures, but there's quite an infrastructure there as well. So each of these enclosures is surrounded by sheet piling that goes all the way to the mineral, the interface of the ancient lake bed and the bog itself, so that each of these chambers is isolated not only above ground, but below ground as its own component for each warming experiment. Heating is accomplished below ground through resistance heaters penetrating 3 meters deep into the peat from the peat surface. Within the outside of the treatment area, we heat from all the way from the surface to the 3 meters in depth. Within the surface, within the treatment area, it actually is a passive heating, and that we only heat from 2 to 3 meters in depth, and we set up a gradient in this treatment area. And I'll show you what that looks like as far as temperature in a second. So there's actually no active heating here, so there's no hotspot effects in the 0 to 2 meter study zone of the system. So it doesn't matter how close you are to these heaters, it should be a fairly consistent temperature. Wait, wants to flip slides. So the SPRUCE experiment sort of began to be conceptualized and do some pre-treatment characterization over a decade ago now, when we first started studying these systems. And the construction, the actual construction of the experiment proper began around 2012 with the installation of the boardwalks and infrastructure. And then it was staged in construction after that, such that the below ground infrastructure went in first, beginning in 2013 and 14. We had a below ground deep heat only experiment where we were just heating the below ground area without before the chambers were constructed. And we had, and then after that, the chambers were constructed and we began heating the whole above ground and below ground system together, and adding a carbon dioxide treatment into half of the plots in each of those chambers another year after. So full operation of the whole experiment with deep peat, with the below ground heating, above ground heating, and carbon dioxide really began in 2016 and has been carrying on since then through a planned 10-year duration. So what this looks like in the systems as far as that warming. So these graphs are showing the air temperature on the left side, the soil temperatures on the right side. The top plots are showing the first 3 years worth of operational data, basically, in temperature, showing, so you can see the interannual pattern here as far as temperature that's maintained through those 3 years, and the differential among the different treatment combinations. So the 0 degree added plots in light blue are on average about 2.25 degrees different than our 2.25 degree target treatment. So that treatment differential is good and steady, and has been fairly steadily maintained throughout the course of those 3 years and beyond now, actually. So You will see minor variations in that, where often those are not actual changes in the temperature of the peat, but sensor malfunctions that have to be replaced or recalibrated. So, oh, I forgot to do something a while back, but we'll skip that anyway. I did have, let me go back. There are a lot of different sort of neat technologies that are being implemented here, one of which we have, real-time webcams in each of the plots at both the above ground, above the high plot level to see the entire plot, as well as within the, at a shrub level to look at the shrub level layer. And they can monitor the greenness and phenology of these systems as they progress over the course of the year. So, for example, this is a 0-degree plot, picture taken about 2 hours ago. And this is
We currently just see your slides, Chris. I don't know if you.
Oh, you can't see the
No, we just see your slides.
web page. Gets too fancy here. Well, we'll skip that anyway, I guess. So
we can stick the link into the Discord.
yeah, it's in my slides. Okay, we'll go back, and I guess I won't try this one either. So there's a, oh my gosh, why does it skip around so much? Stay. There's a link to here also as well to real-time data that can be downloaded from each of the plots for temperature, moisture, water level, a whole variety of different plot variables that are being measured. So the variability across these peatland sites, and even within this peatland site, is quite large, especially with regard to depth. So horizontal spatial variability across the bog is not as great as the variation that you see in chemistry and microbial properties with depth into the bog surface. So as you go down from the surface of the bog, the temperature, the pH, the redox conditions, and the chemistry vary strongly. And this results in in dramatic gradients in the microbial communities within these systems too. So, for example, in the middle panel of figures on the right, you can see quantitative PCR data from the 0 and 9 degree plots. I believe this was in, this data was taken from 2016, where the fungal qPCR copy numbers, for example, drop off dramatically in the first 40 to 50 centimeters in our samples. And below that, with depth, there's very little fungi. Actually, we're nearing our detection limits in some of these cases. With bacteria, it's a similar pattern, although not as steep as the drop off from surface to depth with fungi. So this is a little less dramatic, but still the same pattern. Archaea and methanogens, on the other hand, seem to peak around the -30 to -40 centimeter depth within the bog. And this is the sort of average water table, the average level to which the water table drops each summer on a natural basis before we started heating them. So this is sort of where the methanogens and archaea peak. And it's such that when you get down deep into the bog, you have actually positive archaea to bacteria or archaea to fungi ratios, where you have more archaea at depth than you have bacteria or fungi. And most of those are Cranarchaeota. So throughout the course of the SPRUCE experiment, we've been collecting metagenomic and other types of data. Our lab has primarily taken charge of collecting the metagenomic data in collaboration with JGI. So in 2015, after the first year of peat only heating, where we weren't heating above ground, We collected 38 metagenomes representing all of the built plots, not the ambient plots that year, but and from 4 different depths within the peat profile. As I just told you, depth is a very important variable. So 4 different depths within each profile, we collected those metagenomes for. In 2016, after the second year of below ground heating and the first full year above ground heating on top of that, we collected another 45 metagenomes that were successful. And in 2018, 4 years after 4 years of heating in the system, we've also collected another 48 metagenomes. The difference here between 2016 and 2018 is really just a few samples. One where we had a failure in the sequencing, and 2 where we had where we didn't have a sample, where we had hit the bottom of the bog before we got that deep sample. And from this, we've started to work with the NMDC to really use SPRUCE as sort of a feature project and bring our SPRUCE data into the NMDC so both we can use it and use their tools, as well as other people may be able to see the value of these types of integrated data sets. So within this project, we've collected a variety of different data, not just these 3 data types, but these are the ones that are within the NMDC. So metagenomic data, metaproteomics data, and natural organic matter chemistry data. And throughout the course of this week, you will be examining some of that. And I am now at this point going to turn it over to Spencer to tell you a little bit more about some of the metagenomic data that he's been analyzing.
That looks good.
I just want to get back to show my video here. Yeah, so I'm Spencer Roth. I'm a postdoc working with Chris at Oak Ridge National Lab. And I want to take the rest of our time to highlight some of the analyses that myself and others working in the SPRUCE collaboration have done with the meta-omic data sets that have been generated from the sites. And so to start, I'm going to highlight this paper that was published last year in June. This was led by Rachel Wilson, and I'd like to acknowledge all of the co-authors on this paper that contributed to this work. And as you can see from the title, they looked at the below ground or the soil responses to experimental warming at the SPRUCE sites. And so they highlight the metabolome, but also included metaproteomics and metagenomics in this data set. And I'm going to highlight a few of the key results from this, but I've also linked to this paper at the bottom of the slide in case you're interested in looking further, because there's a wealth of data that went into this manuscript and analysis. And so the first figure here shows concentrations of pore water CO2 and methane in panels A and B respectively. And what you can see is that with experimental warming, temperature across the x-axis, you can see a significant increase in pore water CO2 from both the surface depths all the way through the peat profile down to 2 meters deep. On the right panel, panel B, you can see that at the 2 surface depths, 10 and 25 centimeters, again, there's a significant increase in pore water methane concentrations with increased temperature at the SPRUCE sites. And so with this increased temperature, experimental warming, we're seeing these increases in greenhouse gas production here. One of the panels that I did not include from this figure also shows that the ratio of CO2 to methane decreases with temperature, indicating enhanced methanogenesis with increasing temperature. So they go further into this study. On the left-hand side is a figure showing the nominal oxidation state of the surface peat dissolved organic matter, and you can see that that declines significantly with increased temperature, indicating enhanced decomposition of that surface DOM. And so these are coming from 3 different time points and 3 different depths near the surface of the peat. On the right is a principal components analysis plot, and this is of an integrated data set, which includes metaproteomics, FTI, CRMS, NMR, GCMS, and lipidomics data. And what you can see is that each point represents a particular site at a particular depth, depth represented by the shape of the point and temperature represented by the color of the point. Across component 1, this axis here, you can see that depth is a significant factor influencing the composition of this multiple integrated data set. And they also found that temperature, which is represented here on component 2 axis, was a significant factor influencing this below ground changes in the soil across that integrated data set of metaproteomics, and looks at the dissolved organic matter as well. And so, like I said, this is just a really quick highlight of some of the results from that paper. Again, it's linked here in these slides. If you would like to look further into it, there's quite a bit of data into it. And they included some metagenomic data in that analysis, but what I have been working on in my postdoc at Oak Ridge has been looking at those metagenomes that Chris highlighted earlier that we've generated across the 3 different years and the multiple depths. And I've taken a metagenome assembled genome-based approach, or a MAG-based approach. If you're not familiar with shotgun metagenomics or MAG-based pipelines, I put together just a very basic slide to describe it. So we, working with the JGI, generate a data set of the short sequence reads from our different depths and different sites at the SPRUCE experiment. After quality control checks, we can assemble them with assembly algorithms to generate longer contigs, and then we can use binning algorithms to bin DNA sequences from these contigs that come from the same microbial genomes. So after some further quality control steps, we can generate a data set of MAGs, or metagenome assembled genomes, that represent complete or nearly complete individual microbial genomes coming from our soil samples. And once we have that data set, we can map back the original short reads to get an idea of the composition of the microbial community across the soil. And we can also put them through databases to look at the taxonomy and phylogeny of these microbial genomes, as well as the functional potential of these microbial genomes. And so a really big strength of this MAG-based approach is that when we have a complete or near complete microbial genomes, many of the functional genes are linked together with one another on an individual genome. And so we can start to investigate potential functions across the SPRUCE sites. We're interested in decomposition of organic matter as well as production of greenhouse gases. And so we can start to look at that across the sites. And so we've generated a data set of 810 dereplicated medium and high quality MAGs from the SPRUCE sites, which are represented in this very busy phylogenetic tree here. A couple of takeaways from this: they come from both the bacteria and archaea, 2 domains of life. You can see that the acidobacteriota make up a large portion of our MAG data set, as well as proteobacteria, desulfobacterota, and then several MAGs from groups that are methanogenic. About 85% of our short DNA reads are mapping to this MAG data set, and that's shown in this box plot here. So you can see at the 3 deeper depths, we have this fairly high mapping of the short reads. And what that means is that our MAG data set is really capturing a large portion of the diversity that we find in these peat metagenomes. You can see that at the near surface depth, 10 to 20 centimeters, we have a lower percentage of those short reads that map to our MAG data set. And this is likely due to the fact that We have the highest diversity near the surface of the peatland, and this is where we're also going to get more DNA from plants and other sources that may contribute to this low mapping rate. And so just looking at the taxonomic distribution of this MAG data set across the sites, this plot is faceted by the 4 different depth increments across the top, and then increasing temperature treatments across the x-axis within each facet. And so you can't really visualize a temperature effect here, but what you can see, as Chris mentioned, depth is a really strong driver of changes in the microbial communities. You can see as you move down to the 40 to 50 centimeter depth below the surface of the water table, where you get this large shift in redox, higher relative abundances of the desulfobacterota groups, which are involved in things like sulfate reduction and iron reduction. And then, as Chris alluded to in the qPCR data, in the deeper peat, we see higher relative abundances of archaeal groups. And so although they declined at a sort of lower rate than bacterial and fungal groups. And so you see this high relative abundance of archaea down at below 1 meter depth. And looking at the composition of the microbial community, you can see clearly in this very horseshoe-shaped plot that depth is a significant factor driving the composition of the microbial community across the spruce sites, represented by the color of each of the points, each point representing one site at one particular depth. And you can't really see the effect of temperature in this plot. It really shows this strong depth effect. But if we look at just the deep peat sites, the below 1 meter, our 2 depths below 1 meter, you can start to see the effect of temperature, which is a significant factor influencing the composition of microbial community. And so again, color of the sites represents the depth, and you can see separation based on depth, but you can also start to see that the open points here, which represent the warmer plots, are ordinating on the left-hand side of axis 1, whereas those points that are closer to ambient in temperature are ordinating on the right-hand side. And we found that temperature is causing a significant effect on below ground microbial communities. And so we've also looked at the effect of temperature on individual MAG abundance and relative abundance. And we've found that in this very busy figure, about 10% of the MAG data set is forming a significant relationship with the temperature treatment. So that could be either increasing or decreasing significantly with temperature treatment. And so a lot of the work that I have been doing is going further into that data set to investigate those responses of individual MAGs across the sites. And so sort of to summarize what a lot of the work we've done thus far shows is that we see increased production of CO2 and methane from the spruce sites, and we're also seeing shifts in the soil metabolomes, proteomes, as well as the metagenomes. And this all indicates that as temperatures rise in response to climate change, there may be an increased decomposition and carbon loss from peatlands. And as Chris mentioned, these ecosystems store a large portion of the terrestrial carbon for the area that they make up. And so we are continuing to work, again, as I highlighted, you can look at functional potential of the metagenomes with our MAG-based approach. And so we're continuing work looking at functional shifts in the below ground microbes. And we're also looking at the response to elevated CO2, which we haven't highlighted yet. A lot of the data was generated before CO2 was added to the plots. And so that analysis and those analyses are ongoing, and we will continue to sample the spruce sites through, as Chris mentioned, the 5 or 10 year mark. And so with that, I want to say thank you for listening to Chris and I, and we are happy to take any of your questions that you may have about Spruce.
Great, thank you so much, Chris and Spencer. I need to, every time Zoom like opens and closes full screen, like my windows move around, so now I have to get myself reoriented. We've had a couple questions come in already, one of which was, have you observed any dominant bacterial phylum at lower depths?
I think across the depth profiles, as you saw in that phylogenetic tree, Acidobacteriota are really dominant. And in that stacked bar chart, even though you saw the high relative abundance of archaea down at deeper depths, that main sort of orange bar was Acidobacteriota, which are really the dominant group across the site.
There was one particular mag in previous years' analyses that was composing up to like 15% of some samples as far as relative abundance of one particular mag in the Acidobacteria. So it's quite impressive sometimes.
Great. And then Andrew Lalu asks, do you see any difference in fungi profiles versus depth?
Yes. So I think Chris highlighted in that qPCR data, you can see a really drastic decrease in fungi as you move deeper into the peat, and it really drops off there towards the surface. And there's a group working and looking specifically with our metagenomic data on fungal, mining out the fungal DNA and working with that. And so Chris may have more to say about that.
I don't have a lot more to say about it right now, but there's another student working with Joel Koska at Georgia Tech that has been working through some of the eukaryotic data within the metagenomes. I don't know if we have any great conclusions from that yet.
I think so.
So stay tuned is what it sounds like.
Yeah.
So from Kayumbe Kim, it's a great experimental setup. Have any SOM molecular comparison studies like SOM degradation been done? Any chance to do collaboration?
Yeah, so we have collaborators from across the country and world. Actually, we have several international collaborators, and we've sort of treated it to some extent as we do a lot of the core research at the site, a lot of different core questions that we're addressing, but wherever there's an interesting environmental question that we're not addressing and other people can address, and there's not a dramatic impact on site operations or making Swiss cheese out of the plots, then we're happy to allow people to come in and do research. So some of that is funded research. They get actual grants funding and are using our sites either primarily or as part of a network of looking at other sites too, or informally, some people are, there's been a couple professors that have done some of their research, their startup package research up there, so there's a variety of different folks that are doing things. SOM in particular, I've been working a little bit on decomposition studies, but the organic matter chemistry, a lot of that, Rachel Wilson, Malak Tafali, and others have been doing it, sometimes in collaboration with EMSL. And it's not really, I think mostly Rachel is doing it these days, but I know Malak is still, she has some new funding to start doing some work up there again, I know.
Great, thank you, Chris. From Paula Martins, I'm curious to ask why deeper microbial communities seem to be more sensitive to temperature increases, and what's the response of methanogens at depth more specifically?
Yeah, so that's a good question. I really was just highlighting that deeper peat subset because we see that below 1 meter, there's not as strong of a depth effect. And so that's why I highlighted those two deep peat samples. in that plot, but we do see just across our full depth profile that temperature is a significant effect. And so I wouldn't necessarily say that it is more enhanced at deeper peat at this moment. And then for methanogenic response, I'm just getting into that, and we do see that there is a depth-specific response with methanogens. It's not quite clear from our MAG data set yet, but we do see a subtle increase in the relative abundance of methanogenic MAGs with warming across the sites.
If anything, I would say there's less effective warming deep in the soil profile. It's been a very slow to develop effect over, where we did see change often in some of those surface layers relatively quickly. Deep in the peat profile has been very sort of resistant to change, but there are effects becoming more and more apparent. And part of that may just be those deepest peat soils have been basically sitting there at 6 to 9 degrees Celsius. They've been somewhere between 6 degrees on the low end in the winter and 9 degrees Celsius on the high end in the summer for thousands of years. And now all of a sudden we've bumped up that temperature by another 9 degrees Celsius below ground. And so, there may not be a lot of capacity to take advantage of that change. I don't know. It's an interesting question to think about.
Great. From Monica Schmoll, for the metagenome data, how deep did you sequence per sample? Millions of reads?
Yeah, so that increased between the data sets from 2015 to 2016 to 2018, and Chris can correct me, but it was about a relative doubling every time. And so our 2018 data sets were sequenced fairly deep. I think like on base pairs, it was 30 billion base pairs per metagenome, which is around, I think, 100, 200 million reads per metagenome.
Great. Do you have an idea of which microbes may be driving the changes in the greenhouse gas production?
Well, I think for methane, of course, methanogens are what are going to be producing the methane across the sites. And so potentially increases in methanogens would be driving that increased methane production, but of course you can have methanotropes that may be consuming that methane that is produced as well. That is sort of what we're getting into with this data set now as well. So
great. There are a lot of really great questions coming through, so I apologize if I miss anything, but the nice thing about the Discord is that they will persist. So just want to encourage everybody to look there, but I'll keep going along. Just wanted to mention that. So go ahead, Chris.
I go back and give a more specific answer on the size of the metagenomes in each of those years. I'm just looking at the data slide I had before. That's not in the presentation, but the 2015 averaged 2 or 5 gigabase pairs of QA/QC data per sample. The 2016, 12.5 gigabase pairs per sample. And the 2018, the figure I have is 32 gigabase pairs average QA/QC data per sample. That may have changed a little bit, but anyway, that's what I have.
Thanks, Chris. Okay, from Anna T, have you looked at organic matter composition, FDIC-RMS, and how that varies with temperature?
That has been done by Rachel Wilson and others. That was not really done by us per se, but yes, those data, and I think some of that data is included in the NMDC data set.
Mhm.
The data set on NMDC includes primarily up to the 2016 data set, whereas the 2018 data set is still sort of being generated in some cases and compiled into the NMDC. So there is, Montana knows all about that. And so it does not, that the 2018 metagenomes and organic matter data and that kind of stuff is not in there yet.
Yep, so another stay tuned, keep an eye on the NMDC data portal and our release announcements, and you'll see when it comes up. All right, from Natalie Delherb, did you notice methanotrophs above the methanogens peak?
I haven't.
Go ahead.
Yeah, go ahead. I haven't gotten deep into investigating that quite yet, but it's a sort of good question. I don't know if Chris has more to offer.
Yeah, you know, our sampling scheme for the metagenomes is not really super high resolution in order to sort of see where those shifts are between methanogenic and methanotrophic processes. We go from like 10 to 20 centimeter depth to a 40 to 50 centimeter depth, but somewhere in there, you expect that there's going to be a shift from methanogenic to methanotrophic dominance. And it's really dependent also though on water table. So as that water table drops over the course of treatment and over the course of the summer, that, you know, where that layer is really changes. So in the +9 degree plots, for example, we had a drought in 2021 and they were dry to a meter in depth, which is just incredible. Where, because the drought on top of the treatment, we had a massive dry down in some of the plots. Whereas other years, the natural water table variation mostly historically across that bog, it averages around, it drops maybe 30 centimeters up and down over the course of a year. Whereas it dropped over a meter last, or 2021.
Thanks. All right, from Miho, have you seen patterns with microbial communities with root depth? Any effect of temperature / depth on root growth, the influence of microbes, AMF?
That's not something we're addressing specifically. And part of the reason is the data set that you're looking at here is taken in the hollows. We didn't really talk about it in the overview presentation, but there's a strong microtopography effect within these peatland systems. So you have what's called a hummock hollow topography. And in the hollows where the closer to the water table down low, you can see you're really close to the water table and there's not really a lot of root formation. In the hummocks, the high parts of the microtopography is where the trees are rooted and where the roots are all above that water table. So the roots, for the most part, are staying above the water table. There are exceptions to that, like sedge roots and other things that go deep within the profile, but most things root above the water table. And our sampling scheme was really designed to go from the sort of average water table level downward, as opposed to sample in the hummocks where a lot of the roots were. But there has been Colleen Iverson and some others have been doing a lot of different studies on the rooting patterns within the hummock and the below ground versus above ground response. Yeah.
Great. From Marissa Duckett, for those deep community analyses, how do you maintain an anaerobic environment for extracting the samples? Is an anaerobic environment needed to preserve the integrity of the DNA? I suppose this would be an important consideration for looking at gas fluxes.
For the DNA sampling itself, it's not. So we take, as soon as The core is taken out from that depth. It's divided, put into a bag and frozen on dry ice and returned to the lab. For the DNA and specifically, it's not so much of an issue. Other people have been doing more process level measurements on methane production from the peat and those types of things. There they have to go ahead, as soon as that core comes up, they have to, put it in a serum vial and degas it. So that it can be a consideration for other things, but it's not so much for the DNA and RNA measurements.
Great. Another one from Paula Martins. Also, can you say if you observed any changes in the metabolic potential of dominant acidobacteria mags with temperature or depth?
That is a good question. We do see some definitely within that acidobacteriota group, certain mags falling out that are dominant at depth. But again, we need to dig a little bit further for the sort of functional potential of those differences. But yes, definitely there are certain acidobacterial mags that dominate at specific depths that may decline deeper or shallower, et cetera.
Even within a mag type, I know that there was in one of the mag types that was dominant that previous postdoc Eric Johnson was looking at once. So there was sort of a hotspot of variation for that mag at 40 centimeters depth, where the other depths were more similar to each other between mags. So there can be variation sort of on a population level of some of these mags as well, so that you could see, especially in those abundant organisms in the system, you can actually start to see population level variance in some cases.
Kind of in a similar setting, would love to hear if your group has observed any increase in proteobacteria with enhanced CO2.
So the CO2 treatments were only implemented in 2016, correct, Chris? And so many, so those 2015 and 2016 metagenomes were generated before CO2. And so we really have the one data set, the 2018 metagenomes that were generated post CO2. And thus far, we're not seeing a very strong effect of CO2, but again, we're just sort of starting to dig into it because we do have just that one newer data set that has been generated post CO2 treatment.
We have some recent decomposition community data, right? That shows
we do see a significant effect in decomposition bag studies and in response to CO2 treatment, though. Yes, that's a good point.
But still not as strong as temperature or depth, right?
Great. All right, we'll take time for this one last question. From Changxi, compared to the 10% of genome changed significantly, how much of the metabolome has changed significantly by temperature?
So there I would point you to that PNAS paper that I shared results from. It really breaks down the FTICR-MS results, the NMR results, lipidomics, GCMS. And so within all of those, I think with FTICR, it was thousands of things were either positively or negatively correlated with temperature, and they start to break that down in that paper. So again, a lot of that was led by Rachel Wilson, and so yeah, I would point to that because across there's quite a chunk that there were significant correlations with temperature.
Great. All right, thank you so much, Chris and Spencer. And thank you to our audience for all the great questions.
Yeah.